Today we will talk about incomplete dominance and codominance. Incomplete dominance, dominance and codominance. These are the exceptional cases of Mendelian inheritance. Okay, so at first we will talk about incomplete dominance, then we will discuss about complete codominance. Okay, so now come to the incomplete dominance. Incomplete dominance. This incomplete dominance can be found in mirabilis jalapa. Mirabilis jalapa. Okay, mirabilis jalapa. This is also known as 4 o'clock plants. And another example of incomplete dominance is snapdragon. Okay, so in both case they have a pure variety, two types of pure variety. ठीक है, one is red color flower. This is one kind of pure variety which only produce a red color flower. Red color flower. And another is, this is also a pure variety, it produce white colored flower. White colored flower. Both are the pure variety. Pure variety. If we make a cross between those pure variety, those two pure variety, then we will get such genetic constituents, that is heterozygous condition. Okay, so in F1 generation all individual will have this type of genetic constituents. Okay, let's put it into the Punnett square. Chalo, now see, this is pure red variety, and this is white variety, and those are the pure. One thing you should know that R is the dominant gene. R considered as a dominant gene, and small r considered as a recessive gene. Dominant and recessive. Because of the dominant gene, or because of this gene, the plant produce red color flower. And if this gene are in recessive condition, they cannot produce red color flower because they don't have such type of pigment, and that is why flower become albino or white. Okay, so now see this cross. This is the parental 1 generation, and this is the pure generation, pure red and pure white. If we make a cross between red variety and white variety, then we will get to F1 generation. Chalo, now put, or one thing you should know, another thing you should know, that is from this pure variety we can get only one kind of gamete, that is R, and from this variety we can one kind of gamete, that is smaller. Okay, let's put it into the Punnett square. Smaller, capital R, capital R. So all individual will have this type of genetic constituents. Okay, R is the dominant gene, you should remember that R is the dominant gene and smaller is the recessive gene. Okay, according to Mendel, this heterozygous condition should produce red color flower, but no, here we can see pink flower. In that genetic constituents, it will produce only pink color flower. So here we can see that red gene, which is capital R gene, which is dominant gene, that cannot suppress the recessive gene completely, or this R gene cannot express itself completely. That is why this condition is known as incomplete dominant condition, and this feature is known as incomplete dominance. Is that clear? Next, we can do selfing. This is the F1 generation, and we can do selfing between two individual of F1 generation. Let's see what may happen in that case. So there is the heterozygous condition. We are taking two individual from the F1 generation. This individual will produce two types of gamete. One is capital R and another is capital smaller. And this gamete, or this individual, also produce two types of gamete. One is capital R and another is smaller. Okay, let's put it in Punnett square. Capital R smaller, capital R smaller. So here capital R, capital R, capital R smaller, capital R smaller, smaller, smaller. So from this we can see RR, that is pure, this genetic constituents will produce pure red flower or red flower. This heterozygous condition will produce pink flower, and this homozygous recessive condition will produce white color. Is that clear? Homozygous dominant will produce red color flower, homozygous recessive will produce pink white flower, and heterozygous will produce pink flower. So from this Punnett square we can see the phenotypic ratio, that is 1:2:1, that is red, pink and white. If you see the genotypic ratio, that is RR is to capital or smaller is to smaller, smaller. So homozygous dominant, homozygous recessive, and heterozygous. If you see the genotypic ratio, then we'll found 1:2:1. So here in incomplete dominance, genotypic ratio and phenotypic ratio both are the same. 1:2:1. 1:2:1. So genotypic ratio and phenotypic ratio in incomplete dominance is always same. So now come to the next point, which is known as codominance. CE. So in codominance, if we before knowing codominance, we should know about ABO blood group. ABO blood group or ABO blood grouping is a good example of codominance. Dominance, and as well as good example of multiple allele. So let's see what is it. If someone has A blood group, that means he or she has A antigen on its RBC. And this A antigen is produced because of the IA gene. In case of B blood group, someone who has B blood group, she or he has B antigen on its RBC, and its genetic constituents will be like this. Someone who has O blood group, that means he or she has no antigen on its RBC, that is I0 condition. So here we can see that IO is the recessive gene. And IA is the dominant gene. And IB also a dominant gene. Okay. So in case of AB blood group, you can see here A blood group have A genetic condition, and it has A antigen on its RBC. B blood group, in case of B blood group, they have B antigen on its RBC, and their genetic constituents is IBB condition, IBIB condition, or BIB-I0 condition. But in case of AB blood group, Now see AB blood group. AB blood group here in their RBC, surface of RBC, they possess two types of antigen, that is antigen A plus antigen B. So their genetic constituents is IA, IB. So here we can see that two types of dominant gene are expressing. So in case of codominance, both dominant gene are expressed themselves. No one is suppressing each other. So here gene IA also expressing and IB is also expressing. Both are expressing. So this condition is known as codominant condition or codominance.